the chair of this symposium to speak on next generation genomics and integrated breeding in legumes. Dr. Rajeev Varshney, please.
Okay, thank you very much for this introduction. So I think that Niels and Helen already have set the stage about the genomes, that what kind of advances are happening in genome sequencing technology in the crops. And I would like to extend the topic of Helen because this was from genomes to the fields, and here I would like to mention a few things about the use of our approaches in genomics and integrated breeding for the legume breeding, what we are doing at ICRISAT. Before that, from more than 110 organizations from 35 countries. Of course, not in all the projects, the different projects, different colleagues are involved. We have been discussing in morning in the talk of Howard Shapiro and other colleagues that, well, the main question right now in front of the scientific community is how we are going to feed the world, especially when we will have 9 billion people in 2050. And all of us are trying to do our bits. So in this context, and again, in morning we had this discussion in context of developing countries. So basically this food is going to be or is already happening, but more serious problem in the developing countries because farmers, the smallholder farmers, they don't have enough inputs and they can, so basically they don't have enough inputs in agriculture. As a result, they cannot produce enough food to feed the household. So they are always in this vicious cycle. So they never come out from this one. So the question is climate change, land degradation, loss of biodiversity, food crisis, energy crisis, population explosion, etc. So there are a lot of issues going on. Now, ICRISAT works on 5 crops. In this presentation, I will be talking only on 3 crops, and I'm not saying that only these 3 crops they are going to feed the world, but we will be contributing in this initiative. So not only the major cereal or other crops, but each crop plays an important role. So I think all crops are having important thing. Let me talk about, and they are very important legume crops in terms of providing the protein in developing countries. If you see in the genome context, then they are not big genome crops like wheat or barley, but they are bigger than rice and Arabidopsis. So like if you can see the 740 megabase per in chickpea, 835 megabase per in pigeon pea, and peanut is quite big. And another thing is yellow tetraploid, 2500 megabase per. Other problem with these crops is that the genetic base is very, very narrow. So basically, breeders have been doing great job all around the world in developing superior varieties, but I believe that now they are reaching close to the plateau because in case if you don't have much variation how you are going to breed further, not only for the productivity, also for the nutrition. Very quickly, these crops, they are very important, and ICRISAT works not only for Asia or India. We are having 6 different locations in Sub-Saharan Africa, so our activities, they are in Sub-Saharan Africa and in Asia, and these 3 crops, they play a very important role, and you can see that if you combine all these 3 together, that about 30 million hectares, and if you see in terms of that value, that how much value they are adding, internationally, this is about $15 billion. But at the same time, these crops are important. You can see the yield in the farmer's field is very low, less than 1 ton per hectare. So again, the big question is how we are going to address those challenges. And drought, the severe constraint. But in addition to drought in chickpea, we have the issue of the fusarium wilt, ascochyta blight. In pigeon pea, we have fusarium wilt, Israelite mosaic disease, water logging. In the case of peanut, I put late leaf spot, rust, drought, etc. So there are a lot of challenges. What we are, my sake, now we are also having this, well, dart markers we had, now even Casper assays by using this platform, and we are doing a lot of work on the high throughput sequencing on HiSeq 2500. So right now, those platforms. So this is the power sequencing and those genomic technology. But sequencing or genomic resources are not going to help alone, but maybe for validation of the gene or validating the function of the genes. If you are talking about improving the varieties, you need to have the phenotyping data in the targeted environmental conditions and field relevant phenotyping. So in those cases, so what we are trying to do, we are combining all these approaches together. So this genomics data, including informatics, and then genetics data, and basically this map and trait mapping, etc., with the different logies like physiology, pathology, entomology, bring all, basically this is the paramedical approach. And we call this approach translational genomics in agriculture, that how we are going to translate this genome information for crop improvement. So this is one of the major focus area for ICRISAT research, at least, And then what we are, so for instance, we are developing the in our gene bank at ICRISAT, we have more than 100,000 accesses. If you see the legumes, more than 40,000 accesses. Our gene bank scientists, they have developed the different kind of collections, mini core collection, reference set, core collection, etc. Our breeders, they have been developing those recombinant inbred line population, and now the next generation of mapping population, including magic, NAM. Also, we are having the trilling population, and recently the training population, the different breeding programs. So what we do that we are having a lot of genotyping. Nowadays, we are doing sequencing, phenotyping in the field, and then combining these things data together using the different genetical approaches, GWAS, biparental QTL mapping, multi-parent and association mapping, telling, or we are estimating genomic estimated breeding value. So in the end, either we have the marker associated with the traits, or we have genome-wide marker profile, and we are also doing a lot of work in the area of developing decision support tools. So basically for breeders, if you are having huge amount of data, then they are overwhelmed. They need to have the information which line crosses. So for that one, we are working in close several partners, including Cornell. So what we do that we are having those decision support tools, and by using these things, we can predict or we can tell our breeders that these lines you need to take for the next breeding cycle, etc. So those kind of. Now I would like to mention a few examples. So let's talk about the genomic resources. Howard mentioned in morning about the Orphan Crop Consortium. In fact, when we started to work in these crops, I came to ICRISAT in 2005. At that time point in these crops, we did not have much markers. Like in pigeon pea, we had just 10 microsatellites. And then there are 11 groups. So you cannot even think about this construction of the genetic map. Live webcast. So what we have done that we have been working with those partners, and BGI is also a very important partner in this collaboration. So what we did that we developed the draft genome sequence of pigeon pea, draft genome sequence of chickpea. In the case of peanut, we have collaborated with the US-led initiative, which Howard mentioned, and also with the China-led initiative, so we are partner there. But the good news is that we got the genome sequence in all these crops. You can ask, do we have that gold standard genome sequence here? Then I ask the question back, do we really need the gold standard genome for crop improvement? So in terms of genomic resources, we have those reference genome. And nowadays, as we were discussing from morning, that we are moving towards this identification of structural variations. So just one reference genome is not going to help. We are doing resequencing, and I will give some example of chickpea, and we are doing this resequencing business in systematic manner, because of several reasons, cost, technology, people, etc. In the first instance, we sequenced 90 diverse chickpea genotype, which has this. In chickpea, you have two types of chickpeas, desi and kabli. They are like rice, indica and japonica. So we had, and you can see beautiful diverse chickpea germplasm, and there's few lines. So we sequenced at that time point because we wanted to identify the SNPs. Now in the case of chickpea, we have large number, several million SNPs available. We also analyzed 129 elite chickpea varieties coming from 10 different countries, Canada, Australia, Vietnam, Ethiopia, India, etc. So we wanted to understand the genome architecture of those varieties. And then recently we also sequenced chickpea reference set, and this reference set is having basically 300 line, and this set is a representative set of the global composite collection, which includes germplasm from ICARDA and ICRISAT. So these 300 lines coming from 35 countries, and we sequence them from 5x to 10x coverage. So we have sequenced huge amount of this germplasm. In the case of pigeon pea, we have sequenced the pigeon pea reference set, again those 300 lines coming from the 32 countries. Recently in pigeon pea, we also have hybrid pigeon pea, hybrid breeding program with pigeon pea, which is based on the cytoplasmic male sterility. So what we have done that we have selected, or we have worked with some private sector partners. We took the parental lines from all the leading hybrids, and we also sequenced them. Our purpose is to define the heterotic pool, or to move or to enhance this hybrid breeding program in that direction. In summary, we already have more than 1,000 pulses genomes in our hands, and here you are having this chickpea, pigeon pea, etc. But nowadays we are also doing a lot of resequencing. So those days were gone that when we were doing SSR or SNP genotyping on mapping population. Now we are sequencing those populations here at ICRISAT itself. Recently, we also have sequenced magic lines. So this multi-parents advanced generation intercross lines, there are 1200 lines and coming out from 8 founder genotypes, and we sequenced all 1200 lines at 5x coverage. Now we are also moving in the area of pangenome. So basically we are going towards different genera. So like in the case of chickpea, pigeon pea, etc. So these things are going on. I don't have time to discuss these things in detail, but I will touch few points. So after that, now we initiated this initiative last year, and we are having this 3000 chickpea genome sequencing initiative. And out of 3000, we already sequenced more than 1000. Our plan is to finish this 3000 by the end of this year. So idea is that we have taken these 3000 lines. In chickpea, we have 20,000 access. Of course, in the long term, we will be sequencing the entire germ plant collection, but we are moving in systematic manner. So right now we have targeted these 3000 chickpea genome sequencing initiative. Thanks to Dr. Hari Upadhyay from Gene Bank, we are having good collaboration with him. So in this case, what we did that we are having this sequencing with the reference genome, we do those structural variation, etc. But most important thing is to do the phenotyping. Several colleagues are sitting here, and especially Dr. NP Singh from IIPR Kanpur, who is our coordinator in this project, co-coordinator in this project. We have put these 3000 lines at 4 different locations in India. So we are basically generating the phenotyping data. Of course, earlier also we had the data, but now collection of the phenotyping data is very important. And at ICRISAT also we are having high throughput phenotyping platform, and Vincent Wadage will be talking at later stage about these things. So basically this is the plan. Initiative is open. If you are interested to join, please let me know. We will be very happy to have you on the board. Why we are doing all these things, and I will give very quick examples. So now, for instance, the sequencing data can provide different kind of information, of course, to understand genome dynamics. Second is for the crop improvement. If you see the linkage disequilibrium, we have tried to analyze this thing in the different manners. So as expected, this is nothing new, that in elite lines you are having big LD blocks as compared to the land races, etc. And in terms of the Desi and Kavli, so basically Kavli are having higher LD, bigger linkage blocks, but at the same time this indicates that because of these things, you can do the genome-wide association studies relatively with lesser number, unlike in the maize, because in maize, LD decay is very faster. Other thing is we are trying to understand some those selection pressure, because during the course of the breeding, so what happened that this diversity has gone down, and now by analyzing this land races and breeding lines, you can identify the genomic regions which have undergone the selection pressure. And the question is that now, because okay, sometimes in these regions you are having the genes for early maturity, so because breeders, they are very much interested in that one, so they kept on selecting the same region again and again. Second is sometimes you are having the disease resistant genes, but in addition to that you also have the big blocks which doesn't mean anything. So now the question is, during the course of breeding, can you break that linkage drag kind of thing? So those kind of information, they are very useful for breeding. Next thing is that we also compared the wild species versus land races, and again we try to identify those regions which have undergone that domestication thing. And now based on these things, when we develop the advanced backcross population, etc., this is going to be very helpful. So those kind of things we are doing this thing. So in the case of chickpea, we are quite advanced. In the case of pigeon pea, we are moving. In the case of peanut, what we are doing, instead of having this resequencing business, we are doing exome capture sequencing technology, and we are right now we are using this technology on 300 lines on the peanut. That was about genomics. Next thing is how you are going to use this information for the breeding work. In this context, you need to identify the markers associated with the trait. Here at ICRISAT right now we have this precise and high throughput phenotyping platform coming from Vincent Wadage, and then here is the Linkage scan system. But even this is established right now, but even before that, earlier we used to have root trait phenotyping platform where, and okay, let me put like that. So in the, and I will give, drought is having lot of small, a lot of component traits. In the first instance, we targeted root traits, root biomass, root land density, etc. Why? Because of the hard work of physiologists and breeders for several years, they came out with this concept that root biomass, root land density, they contribute for drought tolerance. So what Dr. Puran Gaur, our chickpea breeder, and his colleagues, they did, they developed the mapping population, ICC 4958, 182, and 8261 and 283. And colleagues from the physiology, L. Krishnamurthy and Kashiwagi, they did a lot of phenotyping on this population. We did SSR and SNP genotyping, people like Mahendra Todi and other colleagues from Genomics Center. And what we did that when we did this QTL analysis by using biparental approach, in both population 4958, 182 and 283, 8261, we found the more or less the same genomic region which contains QTLs for more than 20 traits out of 35 traits which we phenotyped, and we started to call this region QTL hotspot. Because, and then another thing is that this genomic region contributes or contains a lot of QTLs which contribute up to 58.2% phenotypic variation for drought tolerance. This is not only came in the biparental population, and another work what we did that we had that reference set, at that time point we phenotyped this reference set for large number of traits and also genotyped with the DART marker, CASPAR, SSR, SNP markers initially. And when we did the GWAS analysis based on that limited data set, we found that some of those genes or those markers which gave very strong signal in GWAS, they were present in this QTL hotspot peak. So this enhances our confidence further. And now what we are doing, colleagues under AISRF project, so some colleagues, so what we are doing, we are trying to clone this QTL region, and we have used the different approaches, again Dave Edwards, Jackie and other colleagues. So what we have been doing, we are using sequencing technology, different kind of sequencing technology. In the first instance, by using GVS technology on this population, we reduced this region quite a lot, and we put about 40 more SNP markers, which used to be very difficult earlier if you don't have the genome sequence. We also use the scheme-based sequencing technology, and that we generated about 53,000 SNPs. And now colleagues in lab like Deepa and Sandeep Kale, what they are doing, we are working on the bin map of chickpea. And after that, we are using the bin map with the phenotyping data. And then instead of QTL hotspot, nowadays we have started to move towards the trait-based QTLs, and we have better idea now that which regions are associated, which genes are associated for the different traits. This work is in progress. Another thing is that in terms of this phenotyping, the reference set, we had this phenotyping for large number of traits. You can see that in different environments, agronomic traits coming from the different locations. And then we also had this resequence data on this reference set of about 300 lines. The preliminary data, we did this GWAS analysis, and you can see there are very strong signals on the different chromosomes for basically seed weight in irrigated conditions, rain-fed conditions, and yield, etc. And you can see here, and this is coming based on the GWAS, based on the resequencing, and you can see several signals for the 100 seed weight days to flowering, yield, etc., which are very important traits for the breeding application. In the case of pigeon pea, we are using the approach what Niels was mentioning a few minutes back, that what we do that we, and there are different people, they use the different names. We use the QTL-seq, some people use POP-seq, some use shore map, etc. But the idea is same. So you take the basically extreme bulks, and we sequence those extreme bulks together with the parent, and then we try to identify the SNP index, and based on those SNP index, you can easily land in that region which is associated with the trait. And by doing this thing, you can identify the SNP, and not only that, you can move at the amino acid level as well, that which amino acid is responsible or there is mutation or what is happening. So in the case of pigeon pea, we have identified some genes for sterility mosaic and fugitive wilt. Another thing is that in the case of pigeon pea, we are also working on the cytoplasmic male sterility based hybrid system. And what we did here, that by we sequenced the mitochondria genomes long back, and after that, what we did that we analyzed those mitochondrial genes, and we analyzed structural variation here by using this agrogel, as well as that basically, well, based on the sequencing, well, so expression variation here by using the QRT-PCR, structural variation by using that's amplicon at sequencing thing. Based on this detailed analysis, we identified some genes, there was NAT7A or, oh yeah, so then, and we think that they are associated with that cytoplasmic male sterility. In the case of peanut, we have one major QTLs for the rust resistance, and which contributes up to 60 to 70% phenotypic variation. We are also doing GWAS analysis by using DART markers and groundnut. We are also etc. They are working on a lot of work in this thing. So what we are doing, one thing is that we are doing this transcriptome RNA-seq to identify the genes for fugitive wilt, ascochyta blight, helicoverpa in chickpea, heat and drought in chickpea, foliar diseases, etc. In addition, we are also doing now gene expression atlas for these three crops. And on top of that, we are also doing some protein-protein interaction, and also that analyzing this protein structure analysis. For instance, here, this was NAT7A genes for the pigeon pea cytoplasmic male sterility, and they are something related to the drought. So we are doing those kind of work. But the most important thing is that even after that, identify the genes associated with the trait, how you are going to use them in the breeding program. For breeding, we have different kind of breeding applications: marker-assisted backcrossing, marker-assisted recurrent selection, genomic selection, and we are using almost all the different approaches. I will give some example of this thing. So for instance, in the case of chickpea, Puran Gaur and many other partners in India, Ethiopia and Kenya, what we have been doing, that we have done this marker-assisted backcrossing for root and other drought tolerance QTLs. I will give an example. JG11 is the most drought tolerant variety. No other variety could beat JG11 during last 10 years in terms of drought tolerance. What we did that we introgressed this QTL region in this JG11, and then after having after three back crossing and selfing, when we had these lines, we analyzed these things at the physiological level. But on top of that, we did that phenotyping at 4 different locations. And we were very lucky and we were very excited to mention that based on this phenotyping data, now we are having several lines which indicates up to 20% higher yield in the irrigated conditions and up to 12% genetic gain or yield gain in that rain-fed conditions. And this is happening on the topmost drought tolerant variety. So what we are doing nowadays that we are introgressing this QTL region in 10 different leading varieties in India and similarly in Ethiopia and Kenya. So basically we are working. In the case of chickpea, we also did this fusion wilt is another problem, and here you can see the susceptible variety which are high yielding but susceptible, they completely die. But when we introgress those regions, fusion wilt resistant segment, you are having very good good yield and good survival and yield. The same thing for ascochyta blight. So now in the case of chickpea, we got markers for several traits and we are using those things in the breeding program. In the case of peanut, Janila and other colleagues, Manish Pandey or so what they did that they have introgressed this rust QTL in ICG with 91114 varieties, very drought tolerant but susceptible. When we introgressed this QTL there and when we did this phenotyping at the different location, Directorate of Groundnut Research is very important partner here. Now you can see this tag 24, this was susceptible variety, this is the yield, and when we have this rust QTL introgression lines, this yield is quite high and we are having similar kind of thing in the other varieties. Now in the case of peanut, we are also pyramiding the different kind of genes. Very quickly in next 2 minutes, what I would like to mention that well, so far what we have been doing, we were using this marker assisted selection, marker assisted back crossing. While talking to colleagues like Roberto Tobrosad, we always talk that we are teaching basically the tip of the iceberg. The real stuff is here. And this can be targeted by using the new approaches like genomic selection. And we have been working with the different colleagues here in different projects. So what we did initially in the case of chickpea, we worked with IRI, Bhardwaj, Shailendra, etc. and ICRISAT. And then we had defined a training population, and this training population about 300 lines, phenotyped at 2 different locations, and also be genotyped with these DART markers, Casper assay, etc. And then we have also worked with John Lukianic from Cornell University, John Hickey from Edinburgh, and we developed a pipeline, and Abhishek Rathore will be presenting about that. But by using this pipeline, you can analyze 6 models together. So instead of having model 1 by 1, and our chickpea stuff provided the information, if you are having all these things together, the desi, kabli and together, then your prediction accuracy is not correct. So in those cases, you need to go to the group wise because structure plays important role. Second is that based on our information, this Bayesian based model, they were found very good. And now we are moving further in the similar way. In the case of peanut, what we are doing that we have targeted a population of 184 lines. And then this was phenotype for the different traits, again breeding related trait. We genotyped these things with the DART marker or so. And then when we did this analysis by using ISMO pipeline, we call it integrated SNP mining and utilization pipeline, which we developed at ICRISAT. And then again, so by using that pipeline, you can analyze the different model, we optimize this one, and then after that, we move further in context of that crop improvement. So in summary, I would like to say that even these crops were used to be called orphan crops. We say that these crops are no more orphan crops, and thanks to technology and partnership that we have been very successful to generate the data. Now we don't have just reference genome, we are having large scale genome resequencing projects, and molecular breeding is becoming routine, but we would like to see its enhanced adoption in developing countries. So this is very important. I already mentioned that collaboration is very broad, and here I mentioned again some organizations, some colleagues who have contributed to this different thing. And of course, some of these funders we have listed here. So we are having this collaboration or support from the different organizations in that project. And in the end, I would say, let's be ready to feed the world in 2050. And also after this session, we will also go for lunch. Okay, good. I would be very happy to answer the question.
Thanks very much, Rajeev. Questions, please. The talk is open for discussion. Dr. Bhatia, please. Yeah.
Varshney, I would just like to ask, is the genomic selection can be applied to the segregating F2 population and to select the homozygotes for the desired traits?
So your question is that whether we can use the genomic selection for F2 population or,
For F2 population, so that you select the best plants on genomic basis and carry them forward.
okay, so this is comment, not a question, right, sir?
Yeah?
So in the case of training population, we are taking in the different set, which is basically representative set of the breeding lines, but you can also take those biparental population, including F2, real population, etc. We have the phenotyping data, you can use those models and then develop the models, and after that you can use them in the breeding program.
Dr. Narayanan, please.
Narayan from Metahelix Life Sciences. I am particularly interested in the hybrid PGNP. You mentioned that NAT7, the mitochondrial gene sequence, was associated with CMS. It was not very clear to me how you arrived at that conclusion. Did you compare the sequences between the A and the B line?
So yes, so what happened that we have done, so this is, we are still in process of the validation, but in the first instance, what we took all the genes and then we compared the different lines A and B at R lines, and we analyzed this thing, even the gene expression at the different stages and also the structural variations. So based on these indications, we found that, well, this gene is quite candidate gene. And what we did that further, that we also compared the protein level and in the different line, A and R line, and then try to see that, well, how things are happening. But I can talk to you in detail because this is the preliminary results. Yeah.
Okay, during lunch, I hope we'll be able to talk to each other. Any other question? Oh, please, sure, go ahead.
Thank you for the amazing work. So I have two questions, if you don't mind. So one is about the selection efficiency for the marker-assisted selection. So do you have any such a data?
So
compared to the conventional or classical
okay,
breeding selection?
so in context of this, if you are talking about this marker-assisted selection or marker-assisted backcrossing in chickpea, what we used in the case of peanuts, so what we had that when we had these markers associated with the trait, and when we make the crosses, so at that time point, this is also very important, because what happened in past, sometimes these markers were identified from some other source, and when people use in the crossing, they are using some other source. So then you don't have those good results. In this particular case, like if we identify the QTL region from ICC 4958 genotype, and when we made the cross with JG11, and then the markers which were linked with this ICC 4958 or the allele what we had, then we use them and then we select the progenies like that. So we use that kind of process perfectly fine.
Thanks a lot. Second one is about this, because your initial presentation, the initial talk you mentioned about high throughput screening, better to go for the field screening,
Yeah.
right? So, but in your QTL hotspot result, mainly I think from the high throughput screening.
Right.
I hope you might have the QTLs or the phenotyping from the field also.
Yes.
I just want to know, you have any compared whether the same QTL regions or something?
Yeah. So this is very good question. So what happened that initially we targeted this population and we were talking about the root related traits. So what we did that we had this phenotyping for the root related stuff, but at the same time we also phenotype the population for yield related traits, harvest index, 100 seed weight, etc. in the different environments. When we did the QTL analysis with the different data sets, and this was lucky situation. I'm not saying this happens every time, because in the groundnut and the thing we did not have, and that's the reason we say that we had a genomic region. In that genomic region we had majority of the QTLs for root related stuff, but the same region also had the QTL for yield related stuff. So in fact, when we are having those MAVC lines, in those cases, so, and one of that another interesting QTL is for the seed weight or seed size. So now what happened that when we are having those MAVC lines, so one of the factor that why we are having higher yield, because this has enhanced the seed size. So we were lucky that, well, we had these QTLs together.
The last question from the lady.
Hi Rajeev, Julie Hopper, ICRISAT. So Rajeev, you mentioned the tilling populations again. So could you tell us where we are on the tilling populations? Are they being sequenced or?
Which
and can you, I don't know, you mentioned tilling populations. Could you tell us more about tilling populations? Because they're very important for testing when you've identified a trait. It's a great resource.
Yeah.
And so can you tell us more about that?
Yeah. So what is happening? So now I'm not sure, but that because I mentioned a lot of stuff. So if you are asking the question in the case of chickpea, then we have this population ICC 495 of 182, and this population has been sequenced. And 283 and 8261, we have done that low throughput sequencing. And I think that in the chickpea, right now we are having only those two populations related to drought. And we also have some posters from the lab, they can see the others.
Okay, I think we can have one more question from Dr. Dhaliwal, please.
You talked about Q2L hotspots. What are their molecular region and their maintenance? Are they near the centromere or near telomeres?
So one thing is that in the case of chickpea, and also I mentioned that we have the reference genome, etc. So we are having just pseudo molecule. We don't have the information in terms of the centromere. So we don't have this idea in terms of linkage group. If you would like to say, then you can just guess that in which region they are. But the second thing is that our feeling is, and again, we are working in the area of QTL cloning, that what kind of genes are present there, etc. My feeling is that maybe in those regions, you may have some transcription factor. And then maybe they may be contributing not directly, but at least indirectly to control the expression of other drought tolerance traits. So that's our feeling. But we will come to know at later stage only.
Okay, then I think we'll have to stop it because we don't want to delay you for lunch. Let's give a big hand to Dr. Rajeev Varshney.
Thank you very much.